4th annual Ed Schteingart lecture, and I'm thrilled to do it for two reasons. One has to do with Ed Schteingart, and the second has to do with Felix Bäuchlein. You can read about both of them in the program, and so I will not read the entire program to you in the next 5 minutes, but I'll say just a couple of salient things. As you know, Ed was recruited to the University of Michigan in 1959 by Jerome Kahn himself, and Ed spent 50 years here at our university, becoming emeritus 50 years later, around 2010. Ed spent his career studying adrenal tumors and Cushing's syndrome, and as you know, he received many awards at the university, including a Lifetime Achievement Award for his educational efforts running our old GCRC and our T32 for the university, for the medical school. It is Ed's pioneering efforts in adrenal tumor biology that really laid the groundwork for our current endocrinology program, which grew from 4 faculty to its current size of over 26 faculty, thanks to Ed Schteingart. And specifically, his interest in adrenal cancer led him to be the physician for Millie Schembeckler when she succumbed to adrenal cancer, and that really got things going in terms of galvanizing support from Bo himself to put some effort into adrenal tumors, the Adrenal Cancer Clinic, and ultimately our endocrinology program. And it's because of Ed that I came to the University of Michigan in 1999, with Ed really as a mentor for me. And it is with that effort that we were able to grow our program. And the first fellow that I recruited to my small laboratory at that time was Felix Bäuchlein, who may be the first Schteingart lecturer who overlapped with Ed and knew Ed intimately in the context of our emerging adrenal program. So it's really fitting that Felix is here today to present the Ed Schteingart lecture. It's also fitting that Ed was hired by Jerome Kahn, and Felix has cloned some of the genes responsible for Kahn syndrome, and we'll be talking about them today. So Felix, I'll just say a couple things, and I'll be brief. The one lesson I'd like to share with all the fellows in the room is never ever be afraid to hire people smarter than yourself. And you should try to do that often. Surround yourself by people smarter than yourself, and in that way you can ride their coattails for longer than they would be able to ride yours. And Felix is really a tour de force. When he came to lab in 2000, he published 7 papers within 24 months and then left really on a comet tail, and over the last 15 years has published over 200 papers. New England Journal of Medicine ×1, ×2, ×3, ×4, Science, etc., again and again, really to the astoundment of the rest of us in the adrenal community. It's really remarkable for me to say that Felix trained with me. I mean, I've really been learning from Felix over the years, ever since he was in my laboratory, and I'm just delighted to call you my friend and my colleague. It's really an honor for me to be able to do that today. Felix will tell you a bit about his work over the last decade or so. He has risen the ranks in Munich, where he quickly assumed the role of professor and head of their amazing endocrinology group with Martin Ranke, who of course comes from the NIH group, the adrenal group there. And Felix has elevated that group to the leading center in the world in adrenal biology that we have just tried to emulate for the last 15 years. And of course, Felix, if you don't know, has also been the lead for the European Network for the Study of Adrenal Tumors, which is the quintessential network for rare diseases in the world, galvanizing support from the European Union, et cetera, as the real tour de force in adrenal biology. And so Felix, with that, I'm going to bring you up here and say thank you so much for coming here to honor our colleague Ed Schteingart with your lecture today.
Well, maybe I stop here. Well, Well, of course, knowing Gary, you do know that you have to reduce this by 50, 75% or so. But I'm flattered anyway. Thanks you. And I'm also flattered to be here, for the Schteingart lecture. When Bill Rainey was asking whether I would give this, I said, oh, well, there's an Schteingart lecture, great. And I am very honored. As I knew him, I met him here on a regular occasion. And I very well remember one discussion that we had on the difficulties to establish an adrenocortical cell line, because that was what he was also quite involved. And I said, why is that so difficult? And he said, I don't know, but it is. And it's true. So we struggled for 15 years to do this, and we just had the other day a paper out where we finally got something similar to a adrenocortical cell line. So it is quite hard, and I did remember this quite well, he was talking about. So it's great to be here again. This is, this is the hot spot for adrenal research. I again realized this morning talking about with many of the postdocs here from the lab, from the Hammer and the Rainey and the AUKUS and other groups. So it's a great place to be here again. All right. Of course, I'm not talking about decades of things, but I try to show you data which are rather recent. And just to start, this is a very recent meta-analysis, not from our group. This has been published in the JCM very recently, where you see that, and you might not be able to see this, but bottom line is primary aldo is quite common. So it boils down here to roughly 9% of all cases with hypertension turn out to have primary aldo. So it's certainly worthwhile to studying this. And it's also worthwhile because, as I guess most of you here in the audience know, that primary aldo is afflicted with a high likelihood of cardiovascular morbidity. So there are two examples here: stroke, myocardial infarction, and atrial fibrillation. These are the patients with primary aldo. These are the patients with essential hypertension, more or less the same blood pressure, and you see the odds ratio to have stroke and so on is much higher with primary aldo. And here's another prospective study looking into the incidence of atrial fibrillation. And you see here the patients with primary aldo, they have over the time a very high likelihood to suffer from atrial fibrillation. So it's obviously a clinical problem which needs to be tackled. Now, this is a very simplified scheme. Again, I guess most of you know that primary aldo comes in like two flavors. One is the bilateral form of disorders, this is the bilateral hyperplasia, and the other one, the unilateral form, the classical con adenoma. And in most of the cases, you do adrenal vein sampling to separate those two types. And of course, it's an invasive procedure, which is by itself quite difficult. But already when you look into histology, and this is just an example of those, you realize that it's not as easy. It's not either or. But so this is, for example, this is CYP11B2 as the marker of aldosterone secretion. This is a clear nodule, CYP11B2 positive. You already see there are some cases where there is some heterogeneity. This is an APCC. These are some foci, and this is maybe even adrenal nodule, which is CYP11B2 negative. And these are some differences in the zona glomerulosa staining intensity. So you see immediately there might be some gradient in between, and it's not quite clear yet, whether there are some overlaps and maybe even some involvement from the bilateral hyperplasia down to the aldosterone-producing adenomas, or is it rather that there's an inactive endocrine inactive tumor, which then later, turns into primary aldo. And quite recently, this is just a clinical observation. What I show here, these are patients with with primary aldo in comparison to normal patients. And what we did here in our radiologist was adrenal volumetry. So quite dedicated on CT scan to calculate the adrenal volume on the left, on the right side. And we see here, so this is in green. So this is a patient with a con adenoma. This is the contralateral side, and this is a patient with bilateral disease. And volume-wise, these do not show any difference. This is on the right side and on the left side, and they're actually all the same. However, if you compare this with normal people, they have no endocrine disorders, they were gender and age matched, you realize that they are smaller. And interestingly enough, also those patients which have an adrenal adenoma on one side, even the contralateral size is larger than those in a control population, indicating, well, maybe there's some overall growth defect in those patients, and they, as some of them actually go on with evolving an aldosterone-producing adenoma. Of course, this is only observational, but it's quite interesting. So if we now concentrate for the first part on this bilateral part, there is a question mark whether, whether there's some ongoing between a maybe more homogeneous bilateral hyperplasia to more nodular hyperplasia. I'm not going to talk about this. The person to talk to is Bill Rainey here from the group. So the main contributions which have been done worldwide really come from his lab. I'm not going to touch those at this moment, but I'm rather concentrating on this earlier part. So the question is whether there are any genetic contributor which might make the patients more susceptible for high aldosterone output. And naturally, there are the studies, old studies on monogenetic disorders. The first one from the Lifton group was the familiar hyperaldosteronism type 1, glucocorticoid-remediable hyperaldosteronism due to a chimeric gene of the ACTH-dependent 11-beta-hydroxylase and the angiotensin II-dependent aldosterone synthase. So this hybrid gene then led to an ACTH-dependent output of aldosterone. Then what really changed the field was this landmark study, also from the Lifton group. This is now 5 years ago, where they characterized by exome sequencing, and this technique by itself has changed the field, as in many other fields as well. They characterized this potassium channel, KCNJ5, being mutated in a large number of somatic cases, but also in rare cases with familiar hyperaldosteronism. And this particular mutation leads to an inflow of sodium, a depolarization of this glomerulosa cell. This leads to an increase of calcium, and the calcium signaling is one of the main contributor to the increase in aldosterone synthase and hence aldosterone secretion. So this was then called familiar hyperaldosteronism type 3. These were the initial cases were younger children with very high hypertension and aldosterone output, but later on also from Europe, it was quite clear that you might find this also in older patients with less severe forms. And then more recently, again, on the basis of exome sequencing, a type 4 and also another form was found, familiar cases in 2 genes, which I come back to later, CAGNA1H and CAGNA1D, and some of those have also a neurological phenotype, that's why it was called PASNA. Then, of course, you wonder if there's a type 1, 3, and 4, there must be a type 2, and there is, but this is a less well-defined form. This is kind of the remaining cases which are not type 1 or the others. And it has been the question is what underlying genes can contribute to FH2. And I'm just going to show you in the next couple of slides an example how those family can look like, and that it's not so easy to get those to the genetic point. So this is a family that we were contacted by that started with a monozygotic twin here, 2 sisters who had primary aldo. There was a strong history of problems from cardiovascular disorders, likely also primary aldo, but they were deceased and there was no information. And there was a son here from the first sister who was also afflicted, and others had maybe some very minor hypertension. So we did some targeted genetic evaluation. We screened for FH1. There was, but also from the phenotype, it was quite clear that it was most likely non-glucocorticoid-remediable hypertension, and this was excluded by PCR. We screened for mutations in KCNJ5, and we screened also for FH4, and we first looked for the hotspot mutation, which is usually found in CAGNA1H. And to our surprise, we found another variant here in this position in CAGNA1H. We thought, well, okay, case solved. That's easy. Let's look for the other family members. So it was present here with the stars. You see it was present in 2 of those twins in the affected son, but also it was found in those 2 daughters, which had not really, at least not a clear phenotype of primary aldo. But still it could be, you then start negotiating with yourself. Maybe it's not complete penetrance. This has also been found in other cases. Let's go on with some functional studies. And we did in collaboration with Ute Scholl, who is actually otherwise the person who did clone the CAGNA1H and CAGNA1D, or it saw the association with family hyperaldo. And what you see here easily, there's no difference. And we see here, so these are some studies from transfected HEC cells, patch clamping, and this is in vitro studies. This is aldosterone in this originally found variants of CAGNA1H, and this is what we saw, and there was no difference with wild type. So, okay, well, going back to exome sequencing, now we did all the targeted what we did, and we did exome sequencing. We found CAGNA1H again, and we found an RMC5 mutation and a DUC1 mutation. So RMC5 had been described before in African American, being maybe a susceptibility gene for primary aldo, probably damaging. And DUC1 has also been described. This is part of a zona glomerulosa gene, which has most likely some functional consequences. So what we can say is that, yes, this is the first case of an RMC5 variant predicted damaging in PA patients with non-African American origin, but whether or not this is really the only and single contributor to this phenotype still remains open. And this is probably why there aren't, at least for now, not that many cases where there is a clear indication of a genetic or genotype-phenotype correlation. So it gets actually pretty complicated at some point to really make the point that this is the underlying gene. So what other possibilities are there? So one study that we did a genome-wide association study, and here we did not concentrate only on patients with primary aldo, but for the whole range of aldosterone-renin ratio, which I guess you know is the main screening parameter of primary aldo. So this was done on roughly 1,800 subjects. This is a population-based study which is done quite in the vicinity of Munich and Augsburg, a small city there. And what you see here in this Manhattan plot is this significant hit here on chromosome 5. And if you rather screen in, you find a number of genes. One is here an SLC26A2, and we excluded other candidates. And this is a solute carrier which has otherwise been found to be involved in sulfate chloride exchanger, most likely is important for proteochloroquine sulfation, and it has also a cartilage phenotype. There are some patients which have a mutation in this gene, but there were no information about the regulation of aldosterone secretion for this gene. I'm not going to show you all the data, but just one. This was a knockdown study in H295 cells. So we knocked down the gene and looked for the phenotype. And this is just one example where we saw an increase of calcium signaling, but we also saw an increase in the downstream targets here, chem kinases, NUR1, NGFIB, and also CYP11B2, which was upregulated. So at least we could show that downregulation of this gene leads to a phenotype which has some reminiscence for primary aldo. And I took out the slide just to make it a bit simpler, but we also looked into a mouse model, and the knockout of this gene also leads to a phenotype with high aldosterone levels. So again, as an example, that a gene, which you can find here on a population-based study, can have some impact on aldosterone secretion. Another way to look into this is something that I started not long after I left Michigan. So this is an ever ongoing one of those projects where you think it's never going to end. And I have shown this slide on a number of occasions, and Gary is already yawning. So this is a way to reverse genetics. So you induce on a random basis mutations by the substance ENU, and then you screen for any kind of phenotype that you're interested, and then you go back and try to find out the genetic cause of this particular phenotype. And again, we were interested in the phenotype of primary aldo. So we screened almost 3,000 mice. We had 83 which had initially high aldosterone levels. We wanted to be more strict, so we retested them. There were 11 mice which had sustained high aldosterone, and then we had 9 founders. One founder we lost because the cages actually got flooded and the animal was drowned. One of those points where you think about, is it good to move on with this project? And there were many more. So in the end, we had 8 established line. And I show you at least a few recent data that we had with one of those line. This is line number 4, and you see here the pedigree, and you see immediately all the black ones are those with high aldosterone levels, so you would be quite convinced that there is, over the generation at least, in some of those here it kind of disappears, but you are able to maintain those animals with high aldosterone levels. And then what you can do, you can use nowadays, and when we started this was not available, but nowadays you can do exome sequencing, and you compare affected with unaffected animals. And that's what we did here. And we found a number of mutations, 31. Actually, you would even expect more because this mutagenesis is a quite potent mutagenetic substance which induces lots of point mutations. But on the other hand, over the breeding, you lose some mutations which are not required for the phenotype. So 31 were still there, and it was done in 2 animals. 7 were common in both animals, and here are the genes, and it was quite immediately clear that they were kind of hunched together on chromosome 6, these 3, and on chromosome 8, so those 4. And I'm showing you some of the data, but the weakness of the data as they show them right now, they're preliminary in a way, is that we do not have all the different genes separated, but they are always in some kind of combination, which makes it a bit hard, of course, to look at this. So these are these genes, always wild type mutated, which gives up this, I think, 14 or so groups. And these are the aldosterone levels. They are normalized for males and females because there are some differences. And what you see here is that here the mutated one is always lower than the one with the wild type. So this does not really help. Of course, we would think of to have been induced a mutation which leads to higher aldosterone levels. So this is not very likely to be a good candidate. And there's this other group of genes where, in fact, yes, they are always higher in the presence of those mutations. So that's good. Then if you look for expression analysis in the adrenal, again, you cannot read this, but it doesn't matter. You don't see lots of differences. Again, you see lower expression in some of the genes for this right part of the phenotype. Again, does not really help. But you see for some occasions here, at least significantly higher levels. This is CYP11B2. This is NUR1, for example. This is CYP11A1, so SCC, which is actually lower in all of those cases. Not sure what this means. So again, though there might be a difference, and then of course you look for how does the histology look like and what is the CYP11B2 staining. And again here in those mutant ones, the overall, if you look at the whole adrenal cortex, the CYP11B2 expression is higher. And maybe interestingly enough, it's not higher if you look only at the outer area. I didn't call this a zona glomerulosa, but rather so we divided the area in outer, middle, and inner. And there is not much of a difference, but the difference between mutant and wild type is rather in the middle and in the inner area, so rather like a zonation defect and not necessarily only an upregulation. So this is another hint how you could go on with further, and you need at some point, go on with further functional studies in this mouse model. All right. So coming to the other part. So this is now the situation where you have a unilateral disorder. As you have one clone, you say, of maybe of cells which have acquired most likely somatic mutations. And again, exome sequencing has done an amazing job here. And this is the whole spectrum, how an adrenal can mutate and get to a particular disease, so like inactive adenomas or somatic mutation for cortisol-producing adenomas. This was what we recently found. It was I'm coming back to this, the catalytic subunit of PKA. Then all the studies from TCGA and the NSAID group from ACC with all the somatic mutations found here. This is the macronodular adrenal hyperplasia, germline mutations in RMC5. Then the old known Carney's disease genes, the regulatory subunit of PKA, and some phosphodiesterases. And this is what we're talking about here, the aldosterone-producing adenomas with a number of different genes to be found. Again, the first one was KCNJ5. This is essentially this is the same mutation which had been found, what I told you earlier about these early familiar cases in FH3. And this is also happening as a somatic point of view in a number of adenomas. And this is a different studies. They end up with a range of roughly 40% of cases being affected by this particular somatic mutation. There are differences between the gender and between some ethnic background. So East Asia has a much higher proportion of KCNJ5 positive cases than in Western Europe and also the US. So there are still some ethnic differences which we not necessarily understand, but we observe them, and there's clearly this difference. So why is this important? So it could, of course, be relevant that you might be able to use these KCNJ5 mutations in some kind of targeted therapeutic approach. They show distinct electrophysiological differences, and some of those might even be targeted by substances which might be able to block only the mutated channels and not the wild-type channels. This could be interesting. So 40% being affected by KCNJ5. There are, of course, more than that, and these are 3 studies which came out in 2 years ago. And what we found was ATP1A1 and ATP2B3, 2 quite related genes of the sodium-potassium ATPase and the calcium ATPase. And another one is this CAGNA1D I was talking about earlier on, CAGNA1D, which was found, again, mainly by exome sequencing approaches, looking into particular patient cohorts which were very well clinically annotated. So in the end, it comes down to calcium signaling. It's either the sodium influx in the mutant KCNJ5 related gene, which leads to depolarization and increase in calcium. The sodium-potassium ATPase probably has 2 mechanisms. One is the lack of pumping out sodium out of the cell, but it's also the influx of sodium into the cell, which is most likely makes this pump actually a channel, which opens it up and sodium flows in. Similarly here for this calcium ATPase, also sodium properly flows in, but also calcium cannot get out of the cell, which increases intracellular calcium. And the third situation here in this CAGNA1D and CAGNA1H is that the availability or the likelihood for calcium inflow is increased. And in the end, what this all leads to is intracellular calcium increase and autonomous, and this is what endocrinology in the end is all about, or in many cases, autonomous aldosterone secretion, which is independent of the renin-angiotensin system. All right, so this is the last count from our either targeted or exome sequencing and immunic cohort. And as I was saying, you see differences from center to center, from country to country. But this is, at least in Europe, pretty similar. 40% KCNJ5, 8% CAGNA1D. Then the ATPases, there for whatever reason in Munich we seem to have those more often. Then I'm going to come back to the catalytic subunit of pKa in a second. There are a few beta-catenin mutations, and there are still a bunch, like 30% of the so-called wild type. Do not find any particular somatic mutation. And why that is hard to say. Maybe we overlook some, but there's also another possibility I wanted to show you here. So this is an example of a KCNJ5 mutated aldosterone-producing adenoma, CYP11P2 staining, quite clear. And there's the so-called wild-type aldosterone-producing adenoma without any staining. And this is actually a common finding that in those wild-type adenomas we find much less aldosterone-producing cells. Well, that might be, but what is somewhat troubling is also if you look into clinical outcome. So these here in dark green, these are, whatever you call this green, these are cured patients. These are patients who have improved after surgery. So either they need less medication or their hypertension is controlled better. And these are failed, so there has been no change whatsoever between before and after surgery. And what you see immediately here in those wild-type cases, there's a higher likelihood for failed surgery, which makes you wonder, of course, whether this has, in the end, maybe the adenoma was the wrong one to be removed. And I should say that in all those cases, of course, they underwent catheterization and they had the lateralization. But even though the lack of CYP11B2 staining and the lack of this, the somatic mutations seem to have some prognostic value. All right, I'm going to show you another rather cases, because when you do exome sequencing on a large basis, you realize there are always some odd cases. And these were 2 cases which in retrospect already looked like that they might also have, at least one of those cases might have a problem with their glucocorticoid secretion. This is the one here below. So serum cortisol after a low-dose dexamethasone test 5.1 and an elevated urinary free cortisol. I must say at this point we did not look really prospectively, and also glucocorticoid hypersecretion. We do so by now, and I'm coming back to this later. So that was already seen before. And then we did exome sequencing, and we realized that in both of those cases there were actually a somatic mutation in the catalytic subunit of PKA. I'm not going to show you the data, but in roughly 30% of the cases with Cushing syndromes, this particular mutation, the leucine 206 arginine mutation, is found and is very likely, or maybe even you could say it's proven now, that it's responsible for the hypercortisolism. So we found this in actually a con adenoma with also some co-secretion of cortisol. And this is another case where we found another mutation in the catalytic subunit, but this was in a kind of an odd spot, which not necessarily looked like it would have functional significance, and we could also show later that it actually had not. But we did also, again, CYP11B2 and CYP11B1 staining. You might not be able to see this, but bottom line, in the patient with these Cushing mutations, we saw always also high, good staining for CYP11B1 being the most, responsible enzyme for glucocorticoid secretion. Why in this other, probably not really functional significant mutations, it was mainly CYP11B2, and this was found here. And what we found also was that in this kind of Cushing adenoma, there was one small part which also showed CYP11B2 staining, and of course it's quite possible that there is some further mutation going on here, which then drove the primary aldo phenotype. And this could be shown also. This is a steroid metabolome analysis, that those two cases were quite distinct from the remaining ones. So, but this leads me over to the last part of the talk. Is that maybe more common, the glucocorticoid co-secretion of patients with primary aldo? This I have shown you before. So the patients have obviously hypertension, and they have an increased cardiovascular risk. This has been shown over and over again. But there are also a number of clinical observations that patients with primary aldo have also a significant higher risk of metabolic complications in comparison to patients with essential hypertension. And this is true for impaired glucose tolerance, type 2 diabetes, osteoporosis, depression, and anxiety. And the question for quite some time has been, and there is always, this has been shown also on a patient-to-patient view, that maybe there is some glucocorticoid oversecretion. And to look into this in more detail, we did this together with Wiebke Ahl from Birmingham and Katharina Lang. She's doing as quite similar to here, to the steroid metabolome profile, and one possibility is to look into the urine with all those fancy metabolites that I cannot really even, they have quite complicated names. That's I can say for sure. And they give you a good overview in the mineralocorticoid, glucocorticoid, or antigen producing phenotype. And this was the initial study. These were roughly 180 patients with primary aldo. We had similar number of healthy controls, which were matched for sex and age. And in all patients, obviously, aldosterone excess was confirmed, also by confirmation test, saline effusion test, what we usually do. And we did adrenal vein sampling, and we did to differentiate between unilateral or bilateral disorder. Urine was collected, and then this metabolome profile was done. And this is what you see, and kind of also what you expect. Here down the group, black means it was a patient with primary aldo, white means these are the control groups. And if you just sort those by the amount of mineralocorticoid excretion in the urine, you're not very much surprised that it gets blacker and blacker the more to the right you go. This is, of course, what you would expect, that patients with primary aldo have high aldosterone output in their 24-hour urine. However, if you sort those by glucocorticoid excretion, the pattern down here does not really change so much. So this is cortisol and this 6-beta hydroxycortisol, And what you see here that is also to the far right side, those patients with high glucocorticoid excretion are more likely to be con patients rather than a control. And this is not really the case if you do this by androgens. Here you see this is more or less a random pattern. Now, of course, you can then sort the patients in control and patients with primary aldo. This more or less shows the same thing. So tetrahydroaldosterone is higher in patients with primary aldo, no big surprise here. But also the secretion of, so the cortisol secretion in the 24-hour urine and the total glucocorticoid excretion, which is a summary of all the glucocorticoids in the urine, they are also significantly elevated in patients with primary aldo in comparison to the healthy controls. Well, this is an observation, and again, of course, you're asking yourself whether this might have any any impact in the metabolic risk profile in patients with PA. And at least what you can do immediately is you look into the clinical phenotype, and you will see here there are, as you can see here, BMI, waist circumference, fasting insulin, OGTT, HOMA-IR, and all the red ones here are significantly correlated, and they are actually normalized by a number of items, including gender and age and so on. So they are mainly and only associated with the total glucocorticoids, while here the tetrahydroaldosterone doesn't really make any difference. So they are only correlated with the glucocorticoid output, and this is also true here for diastolic blood pressure, and the main contributor with regards to the tetrahydroaldosterone is systolic blood pressure. So there seems to be some differences in terms of metabolic and maybe more cardiovascular association, of course. Then, of course, you're asking yourself, how high is this cortisol secretion? If you compare this with a group of overt Cushing syndrome and subclinical Cushing syndrome, and subclinical Cushing syndrome, of course, has a long debate. How do you call this? There has been a recent European Society of Endocrinology and ENSA clinical guideline, which converted those with normal cortisol secretion and those with cortisol secretion which is above the normal range. And there's some which might have higher cortisol secretion. So this we annotated as SC1 and SC2. And if you compare, and then we had a control group which would fit into this category, SC1, SC2, and the overt Cushing patients, and you see the numbers are not so low. Again, tetrahydroaldosterone is only elevated in primary aldo. And if you look for 24-hour cortisol secretion, you see here that the patients with primary aldo are at least in the range of this SA1 and SA2, but of course they are lower than those patients with overt Cushing syndrome. So they seem to be in a quite similar range that those at least with subclinical Cushing syndrome. And the same holds true for the total glucocorticoid excess. They are even a bit higher, the patients with primary aldo, than those with the subclinical Cushing's. If you expect that there is some glucocorticoid output, of course you would ask yourself what happens in those patients who have the unilateral disease and who undergo surgery, and what is their postoperative cortisol reserve, because you would expect, at least in some of the patients, maybe they develop actually adrenal insufficiency. So these are the baseline values. After surgery, these are, again, the controls in the patients with surgery who have undergone surgeries of this con adenoma, and they seem to be lower already at baseline. And after ACTH stimulation, they are also, so as a group, they are significantly lower than the normal control. And as a matter of fact, a number of those actually, so roughly 30%, formally would fail the ACTH stimulation test. Whether or not this is a clinical relevant issue is, of course, a good question, but it seems to happen quite often, and it only fits to the finding that some of those patients have a glucocorticoid output. Well, if you did surgery and it got better, or even they have adrenal failure, then you would argue that the surgery has done something good, but does it really come from the adrenal adenoma, or is it maybe the adjacent adrenal which is doing the cortical secretion? And that was the reason why we looked more into the tumor, and we did this was done on a tissue microarray with in the end, now we have roughly 120 cases or so, and we did CYP11B2 staining, being the main enzyme again for aldosterone secretion, CYP11B1 mainly for cortisol secretion, and we did some comparison in those with high and low glucocorticoid secretion. And what you see here already is that CYP11B1 correlated significantly with glucocorticoid output, but not with tetrahydroaldosterone, and vice versa. CYP11B2 correlated with tetrahydroaldosterone, but not with glucocorticoid. So this fitted quite well, and as these were punches taken out of the tumor itself, it looks like it's actually the tumor which makes also glucocorticoid. And this is just to illustrate this a bit more. So this is a patient out of the group with high glucocorticoid secretion, which has higher CYP11B1 in comparison to this one with low glucocorticoid excretion and hardly any CYP11B1 staining. And there are nice, fancy ways to look into this into more detail. So there are these color-coded figures where you get a number of from immunohistochemistry, but also from morphological data out of this, which can be quite well quantified and then put into context with the glucocorticoid secretion. And something rather for the future, what we are doing right now, we're using this TMA for a nice technique. This is a multi-mass spectrometry imaging tool where you can do metabolomic studies within the tissue. And of course, it's nice to compare then aldosterone and cortisol-producing adenomas, but even in the group of aldosterone-producing adenomas, those with high secretion and low secretion of glucocorticoids. And just to give you an impression here, so this is CYP11B2, this is more or less the same. Here, this is a little bit higher, again, CYP11B1 versus here. But if you look at some of those, and these are only a few candidates out of a few hundreds, those candidates of the metabolome profile, you see immediately quite dramatic changes actually here in hexose, phosphate, and there are some metaboloids which are not really annotated yet, so we don't know what they are. But as you see here and vice versa down here, AMP and another one here which is much higher in this group. So that's quite interesting to dig into further details and do the clinical annotation and the phenotypic annotation, and maybe to fill the gap between genotype, phenotype, correlation, and to look into more in more detail. And we're just starting to look into those studies, and these are clinical endpoints. You're not going to be able to read them, it doesn't matter. So the outcome data and whether the patient had low or high potassium and stuff, things like that. And these are metabolites which are significantly correlated. These are data from the real morphometry. So if you look at the morphological data in a more quantified matter, you can get lots of information. And this is from immunohistochemistry, CYP11B1, B2, 3-beta-HSD staining, and to correlate this with clinical outcome, which might be interesting in the future. So that was the outlook. What is remaining to say that I have to thank many people here in Munich from the lab and also from the con registry. This is something else which of course we're feeding into quite a bit. All the NSAID group, and there is an ongoing NSAID-HT study which is looking primarily into endocrine-related hypertension. Many people from different centers and countries. And I thank you very much for your attention. Thank you. Am I supposed to say, yes, please, Gary?
I guess I'll take the prerogative if I introduced you. Wonderful, Felix, really connecting the past to the common genetics and metabolomics and where we're headed. The question I had has to do with how your data may lead us to understand one thing Bill's interested, lineage, cell of origin of hyperaldo, and two, the proliferative defect, and how these mutations actually affect cell cycle. And if you can give us any hints as to where that's headed in terms of how these calcium perturbations may tell us about cell of origin in the glomerulosa fasciculata cell and or activation of cell cycle.
Right. So in terms of the lineage, of course, I mean, this is the problem with these clinical data. You have the tumor as it is, and you don't have any information how they got to the point where they are when the surgery had been done. So there's no way of lineage tracing or these kind of things, and whether they had at an earlier time point maybe an inactive adenoma or only glucocorticoid secretion or vice versa. So patient history and these kind of things probably not going to help. So in the end, you have to go to do what you do, and to look for most likely in vivo studies that really give you a hint in terms of, what is there a common lineage, or is that something which goes in parallel or so? So what we see in these mutagenesis-driven animals, it looks like that there is also a zonation defect, which had been shown also in other knockout animals, the task 1, task 3, for example, where they showed that actually the zona fasciculata is full of CYP11B2 expressing cells, and all the zona fasciculata all of a sudden makes aldosterone. And this looks a little bit, certainly not as black and white in the animals that we now have generated by random mutagenesis. And with regards to the cell cycle, I don't have any further insights. All we know is that, for example, KCNJ5 mutations in vitro seems to be bad for the cell, if anything. So you have a hard time to keep them alive and to maintain them, and certainly not that you're switching on cell cycle and they get really a tumor. It might well be that there is I still think that there might be a nodularity hit, and another one which is more affecting the endocrine phenotype, but this might again be too simplistic. Bill.
Two questions about what I think is very exciting, the potential association of subclinical nutrition with primary element. So the first question relates to the fact that you suggested that it's probably tumor related. And I probably missed this, you probably had it, but the bilateral patients with bilateral based PA did not have an elevation in cortisol?
Well, yes, they did. So you're right. So it's not that easy. So in the beginning, the first part of the patient, the group of patients, they were combined, and they did not really show a difference between bilateral and unilateral. Actually, when we started this, we thought, well, maybe this is a good way to do differential diagnosis. You do the urine, you do a CT scan, you see, okay, this is the pattern for your unilateral disease. You see it on the right side, take it out. Don't need catheterization. It's not going to work, at least not in our hands. So the markers are not really helping with regards to differential diagnosis, which means on the other hand, yes, there are this 30% of patients with bilateral disorder who have also the glucocorticoid secretion. And those, well, we do not have any follow-up, no surgery. Well, we do have follow-up, but we have no surgery, and it might be that they have small nodules, which are, I don't know, but you just don't know. But for those who have undergone surgery, all we can say is the surgery did help. So it seems not to be a clear bilateral disorder, which is, only half of the adrenal has been taken out, and this helped. That's I kind of doubt, but who knows? But we can also say that by immunohistochemistry and so on, that at least the correlation of the expression within the tumor correlates quite well with the endocrine phenotype. It does not exclude the possibility that also the remaining adrenal is contributing to the glucocorticoid secretion, but at least it looks rather like it's the tumor which does both of it.
And the other, because obviously you're in somatic mutation so much, was there any somatic mutation that correlated with cortisol?
No, not really. Not really. If you're looking at now from those metabolomic studies, it looks like that the genotype, there is some genotype-phenotype correlation. And what I would find interesting or curious is a bit, all those have increase of intracellular calcium, and they switch on CYP11B2. So in the end, you would say it doesn't really matter, what kind of mutation leads to this. But with regards, for example, to those 18-oxosteroids, it does. KCNJ5 patients, as you know very well, have much higher 18-oxocortisol levels than the other remaining ones. And you wonder, this is not just because calcium levels are elevated, but it's much more likely that it's a lineage problem. They appear in another compartment of the adrenal or something like this. And in that regard, I would be curious to see whether there are any metabolic changes which are rather related to KCNJ5 rather than to the remaining part. And that's what I kind of hope to, as I was saying, to fill the gap between the genotype and the phenotype. Yes, please.
Okay, so as we have learned over the past years, the
Mhm.
And you also show that in those cases it was more likely for those not to repair surgery.
Mhm.
So did you examine carefully every block to see if the PA seating was outside of the supposed
Right.
This is the first question, because we oftentimes ask for pathology blocks, but we might not have access to all the blocks.
Yeah.
So if it's not in the adenoma, maybe outside.
Mhm.
Some other possibility, And do you also routinely check everyone for cortisol secretion as well? Because could the ABS, as you said, they lateral bias?
Yeah. Yeah.
Or are we not?
These are both good points, and maybe I just read it. The first question was, so in a way, maybe we just missed something, and we just looked at one part of the adenoma, and we missed the CYP11B2 stained remaining part. And this might well be true, actually. So it might be a sampling issue for our lab, because the remaining adrenal went to pathology, of course, rightly. And that's true. But what is a bit, as I was saying, a bit worrisome is the clinical outcome, which seems to me more likely when you remove such an adrenal, tumor-carrying adrenal, that the likelihood for cure at least is less than in the remaining ones. This might still be a bit of a random thing, and it's certainly not black and white. But it looks like that there's also some clinical consequences for those particular patients, and you wonder why that is, because the lateralization were in both of them were the same. So it means that at some point within the adrenal, some aldosterone had to become, get out of those, which were more than on the other side. But still, these are, our data. But I agree that it might well be a sampling problem, and that this, piece of tissue that we looked at was by chance just a. Absolutely, you're right. No, might be true, and we might have missed another part of the adenoma. So it could be pretty, a heterogeneous tumor, and the part that we were looking, do the staining and then do the genetic annotation, and that we had, by bad chance, we just had a corner of the tumor which were negative for both. And with regards to glucocorticoid secretion and the interpretation of the AVS, it's true. You could consider if a tumor is secreting also at least large amounts of glucocorticoids, and you normalize by cortisol, that this might screw up your lateralization. That's true. So if anything, you would underestimate the aldosterone secretion, and if it's still, you had the lateralization. That's right, and this could still happen. I do remember one case where there was a glucocorticoid, so a subclinical, cortisol secreting tumor on one side and the conadenoma on the other side. But these are very rare, I would say. And in those cases, you would underestimate the aldosterone output, which was, as it looks like, still high enough to convince us to do surgery. So, but you would have to look into those where you did not do surgery, and those might still have a co-secreting aldosterone cortisol secreting tumor. That's right.
All right, I think what we're going to do is thank Felix, and the division and the Schteingart family would like to invite you all to a reception directly following out here to honor both Ed Schteingart and our guest lecturer, Felix Boischlein. So thank you, Felix.
Thanks.